So we're beginning chapter 3 in the Pierce book, and the reason that we are went from high chapter numbers, so 10, 12, 13, and 15, and now we're wrapping back around to chapter 3, is because we're actually switching disciplines of genetics. So our molecular genetics section had everything to do with how DNA was made up from a chemical standpoint, how it was replicated, and then how protein was expressed. So that's why we spent so much time the first part of the semester talking about how chromosomes vary, how DNA is actually copied during the S phase and it replicates, so it's ready for meiosis and mitosis. And then we spent the last 2 weeks, actually last 3 weeks, talking about transcription and translation in chapter 13 and 15. So that is our molecular genetics section. And now we're switching gears and going back to basic principles of heredity. And we're looking at transmission genetics specifically. So transmission genetics is a discipline of the genetics field that really is associated with how genes move from one generation to the next and how they're expressed. So, we're going to start by looking at basic principles of heredity. And the reason that we have left transmission genetics to the end of the semester is because these concepts tend to be a little bit easier to grasp. And whereas molecular genetics has a lot of moving parts and pathways and upregulation and downregulation and all of that good stuff. So I have found that concentrating on the moving parts of molecular genetics in the middle of the semester tends to stick a little bit better with students than hitting that at the end of the semester. So we are going to hit transmission genetics at the end of the semester because students tend to have a better time grasping those concepts. All right, so chapter 3 is going to be completely different than what we've been doing the last couple of weeks. So we're going to kind of take things back and start reviewing principles of heredity. We're going to talk about monohybrid crosses and how Mendel had done a bunch of work to determine the principle of segregation and then the principle of independent assortment. And he used both monohybrid crosses and dihybrid crosses to actually um, write and author those principles. So within monohybrid crosses, we're going to look at dominance and recessive traits, genotypes versus phenotypes, and then we're going to spend quite a bit of time talking about probability and how to predict outcomes if certain genotypes are paired up. Then, toward the end of this lecture, we're going to talk about dihybrid crosses and how the principle of independent assortment actually came about. We're going to look at less complicated way to actually predict probabilities and outcomes. And then we're going to actually test a couple of hypotheses to make sure what we're observing in a data set or in the real world is actually reflective of Mendel's principles or not. All right, so this chapter is really all about the principles of heredity, so how genes are passed from generation to generation. So the example that the book started out with was um, with the Solomon Islanders in the Pacific, and these islanders tend to be have pretty dark skin and dark hair. However, there is a large population of the islanders that actually are born with blonde hair. And this trait really traces back to a one gene um, mutation that has been populated over time and has been able to work its way from generation to generation so that now we have a large percentage of these individuals within this culture. So that's just one example of looking at how genes transmit from one generation to the next. First and foremost, in order to get a good grasp of Chapter 3 material, you need to make sure that you review the terms shown on this slide and have a really good concept of what they are. We mostly talked about these terms in Chapter 2, at the end of Chapter 2. So we're going to be talking a lot about genotypes and phenotypes, so please make sure that you have a really good handle on what those are, as well as genes, alleles, loci, the difference between a heterozygous genotype and a homozygous genotype, and then what a characteristic or a character actually is. So just to review a couple of concepts, you guys should already have a pretty good grasp of this, but it's important to review it as we set the precedents for Chapter 3. So, as you may recall, genes actually exist in different versions, and those versions are called alleles. So, an allele is an alternate form of a gene. So, a gene is going to be a location on a chromosome that codes for a specific trait, and that the specific version of the gene or the allele may encode for one trait, whereas another version of an allele encodes for another trait. So, the example that we're going to talk about quite a bit in Mendel's work in Chapter 3 is round versus wrinkled pea seeds. So, in this example, the big R allele actually codes for round seeds, and the little r allele codes for wrinkled seeds. So, these different alleles can actually appear for a particular gene trait, and they can actually occupy the same locus on a homogeneous chromosome. So, just keep those basic principles in mind. So the father of genetics is Johann Gregor Mendel, and he really did a bunch of his work in the mid 1800s, and he is responsible for um, the whole discipline of transmission, or what we call Mendelian genetics. So Mendel was born in the Czech Republic and actually grew to be an ordained priest. He actually lived in the monastery, and he was, during his time in the monastery, he was a teacher of many different subjects, everything from physics to botany and plant physiology. And the work that Mendel did, which we're going to go through in subsequent slides, was really ignored for a large percentage of the later part of the 18th century, about 35 years. And so those he still is considered the father of genetics, but realize he did a bunch of work in the mid 1800s, and then his work wasn't recognized for about 35 years later. So Mendel's laboratory was really the garden at the monastery, and he used garden peas as his plant model. And the reason that he chose peas was, first of all, it made a great model organism. It completed one generation in a single growing season, produced lots of offspring, and he had the ability to control self fertilization. So he was able to remove immature anthers before pollination could actually take place. In addition to those characteristics, peas were also a really good model organism because there were quite a few varieties that were available, which allowed him to study how different traits were inherited from one generation to the next. And what really set Mendel's work apart from previous work is that he focused on very simple traits. So he focused on white versus gray seed coats and round versus wrinkled seeds primarily. So, up until this point, there were plenty of scientists that were actually trying to study um, how traits were passed from one generation to the next. But Mendel did something a little bit different that helped him to make really prominent conclusions about his work. So, first of all, he quantified his experiments. That means that he didn't just run an experiment once, he ran it many, many times. Mendel also went on to formulate hypotheses and imply the scientific method instead of just making observations and assuming that those were fact. He was a true scientist in the fact that he had OCD. He paid very close attention to detail and recorded everything. And then he actually took his work a little bit further and did some data analysis, which really hadn't been done up until this point. So he determined numerical relationships and recorded and then computed ratios of the offspring. So the phenotypes that Mendel focused on were seven, and So those different traits are shown on the slide. So everything from seed color to shape, seed coat color, the flower position, how long the stem was, and as well as the pod shape and pod color. So very simple traits, easy to designate. And what he ended up doing in his outdoor laboratory is he worked with 34 true breeding strains of peas, and he actually spent 2 years selecting the varieties that he was going to use. So just to make sure that they were pure or bred true. And what that really amounted to was that they were homozygous in nature. So he used monohybrid crosses as a first step in determining um, the principles of genetics and how genes and traits actually flowed from one generation to the next. So these monohybrid crosses actually were looking at a single characteristic. So one of the ones that we're gonna talk about pretty in depthly is the seed shape. So round versus wrinkle seeds. So he was only looking at one trait at a time to determine exactly how that trait was inherited from one generation to the next. So he did these monohybrid crosses, and he had really tight control over them because he um, actually had the ability to prevent self fertilization. He would go in, remove the anthers, and then he would dust the stigma with pollen from another plant that was carefully chosen by him so he could accurately test the cross. And then he used that pollen to fertilize the ova within the flower, and all of the offspring would then develop into seeds. And from each seed, they would develop into a plant. So, this is how Mendel really controlled exactly the crosses and allowed him to study the inheritance of very specific traits. So, we'll pick up with monohybrid crosses in the next segment.